Hi folks, welcome to the first lesson in the Advanced Higher Biology course. We're going to kick off with Unit 1 and with Topic 1, which is Laboratory Techniques for Biologists. So the first thing to talk about, it's not the most exciting thing in the world, but the first thing that we need to talk about is health and safety. So we need to be conscious that substances, organisms and equipment in a lab can present a hazard. Hazards in the lab include toxic or corrosive chemicals, heat or flammable substances, pathogenic organisms and mechanical equipment. And most of them probably, they make sense, aren't they? There are a lot of them are common sense there, but if it's got one of those toxic or corrosive labels on it, you're going to have to be conscious that it could be a hazard. If the substance that you're using can catch fire easily, then again, we have to be conscious of that. If we're working with microbes, for example, that have the potential to escape from the agar plate that you're using, again, that's an issue, and we have to be conscious of how we manage that using things like aseptic technique and mechanical equipment as well. So something like a centrifuge, for example, is obviously something that could potentially be dangerous if we do not use it appropriately. So all of these things are potential hazards within the lab. Now, hazard is often associated with risk, okay? And what we have to do is ensure that we minimize the risk within the lab work that we do, and we do that through the use of something called a risk assessment. So first of all, a definition of risk. Risk is the likelihood of harm arising from exposure to a hazard. Okay, good idea to remember that one. Okay, it's the likelihood of harm arising from exposure to a hazard. So for example, if you are working with ionized water, the risk is relatively low because if you're exposed to that water, it's not going to be as bad for you than if you were exposed to hydrochloric acid or nitric acid or something, something else like that, something nasty. So you have to be conscious of the risk when you're doing various things. Similarly, in the biological field, if you're working with live organisms, there's obviously a degree of risk there. Bigger organisms obviously have the risk of becoming physically involved with you, whereas pathogenic organisms have the risk of infecting you, via your respiratory pathway or something similar. So we have to always be conscious of that. The risk assessment that we have to write when we assess that risk involves identifying control measures to minimize that risk. Okay, and you can see a few of them on the screen here. Things like wearing safety goggles, protective clothing, protective masks, gloves, etc. All of these things will help minimize some of these risks when we're using some of these various hazards within the lab. There's a nice list there as well at the bottom of control measures, appropriate handling techniques. So, the way that you move things around, the way that you pick up and transport various things within the lab, protective clothing that we just covered, and equipment, and obviously aseptic technique, which I mentioned a moment ago in association with the pathogenic organisms. And we'll talk a little bit more about aseptic technique towards the end of Unit 1. So the next part, it talks about liquids and solutions. Okay, methods and uses of linear and log dilution. So if you were asked to do a linear dilution, you would be looking to create a series of dilutions which went up at a regular interval, something like 1, 2, 3, 4, 5, or 0.1, 0.2, 0.3, 0.4, 0.5, 10, 20, 30, 40, 50, that kind of thing. Okay, that would be an example of a linear dilution series. Whereas a dilution in a log dilution series differs because it's going to increase or decrease in a constant proportion. So for example, 10 to the -1, 10 to the -2, 10 to the -3, and so on. So usually, by a factor of 10 each time, which means obviously exponentially we're going to get a greater increase in dilution with every dilution that we do compared to something like a linear dilution. Okay, so just keep that in mind. If you are diluting by a factor of 10 like that, or something similar, a constant proportion, then a log dilution would be the one you're doing. And if it's in a sort of a sequential, in a series, then it's equal intervals like 1, 2, 3, 4, 5, or 10, 20, 30, 40, 50. We're looking at a linear dilution for that one. We also need to know how to produce a standard curve to help us determine an unknown. Some of you might be doing this for your project, or you might be doing an experiment in class relating to this. Basically what it involves is plotting measured values for known concentrations to produce a line or a curve which allows the concentration of an unknown to be determined from the standard curve. So you plot, like at concentrations that you've made up and you already know, and you plot them against another value. In the example graph here, it's absorbance. So we'd know the concentration, and we'd then measure the absorbance at that concentration. Then what we do is we get something where we don't know the concentration, and we do the same test on it, and we'd measure the absorbance. And depending on where that absorbance value lay along the line or the curve, that would determine the concentration of that unknown. Okay, so that's the use of a standard curve for us in biology. Other things we use in experiments in biology, things like buffers, for example, if we want to keep a constant pH, so perhaps we're adding something to our solutions and we don't want the pH to change, we don't want it to become an additional variable, a confounding variable in our experiment, then we would use buffers. And what that a buffer does is basically when you add acids or alkalines to a buffer, it has a very minuscule effect on the pH overall, okay? And it allows the pH of your reaction mixture to be kept constant throughout. And you can see from the little diagrammatic version on here on the right, we've got, for example, we've got pH of 7. When we add water to it, we see, so when we add acid to it, we see the pH obviously going down, becoming more acidic. But when we've got the water and buffer together and we add acid to it, we see there's very little change in the pH. Similarly, we've got water here, we add the base to it, it becomes more alkaline and more basic, and water and buffer and similar. There's very little change in the pH, okay? So the buffer helps to keep that pH constant throughout the reaction. Method and uses of a colorimeter to quantify concentration and turbidity. So a colorimeter is, again, a common piece of equipment used in 6-year advanced higher projects. You probably all already have used it perhaps in chemistry as well at some point, if you also do chemistry. Basically, a colorimeter will it was actually 2 ways to measure the solutions that we might place into it. But first of all, before we actually measure anything, the first thing we need to do with a colorimeter is we need to calibrate it so that the values are at 0. So we calibrate it with an appropriate blank as a baseline. Quite often water, just plain water would be fine there. And then we'd use absorbance to determine the concentration of a coloured solution using suitable wavelength filters, okay, red, green or blue usually. You can see the button actually on that colorimeter on the screen where you can change between the wavelength filters. And then we can also use teach transmission to determine turbidity, such as when we're looking at like cells in suspension and it's not just a coloured solution. So we can use either absorbance or transmission depending on the circumstance there. Okay, and we would calibrate our colorimeter at the start with an appropriate baseline, an appropriate blank, sorry, for a baseline, something along the lines of water or something similar. Another separation technique is the use of a centrifuge. I mentioned it earlier on as perhaps something that could be a potential hazard, and that's because when you place your tubes into the centrifuge, it spins very, very, very fast. Almost unbearably loud as it spins stuff around, and that's even with the sticky pads that hold it on the lab bench. But the centrifuge, what is the purpose of it? Well, it's used to separate substances that have different densities. Okay, so the more dense components end up settling in the pellet, and the less dense components remain within the supernatant. Okay, now if we have a look here, we've got a diagram of some blood. You can see the plasma, the white blood cells and the red blood cells have all been separated there. Now the supernatants, the stuff at the top, the less dense stuff at the top, the pellet is the more dense stuff at the bottom. Okay, so we'd find the pellet at the bottom of our tube when we were doing a centrifuge. Okay, paper and thin layer chromatography. Again, you probably did chromatography in early secondary school and perhaps in chemistry, perhaps in biology to separate different components from a solution. In advanced higher, we need to know about them in terms of separating different substances such as amino acids and sugars, and the speed that each solute travels along the chromatogram. Okay, that's this thing here. Okay, so the speed that it travels all along this chromatogram depends on its differing solubility within the solvent used. Okay, so we'll put our solution down at the bottom here, and what we'll see is that depending on its solubility, each solute will travel further up the chromatogram, okay, or up the filter paper, whatever it is that you happen to be using. And we can see some different amino acids here that have been separated via this method. Now, we can also use something called thin layer chromatography, which is not in the picture, that's paper, but thin layer chromatography is where we get something like an absorbent material like silica gel, and we place it onto like a glass sheet, and we use that as the means by which to separate our amino acids or sugars. And again, it works in the same means that they move towards the solvent front at the top, away from the solution at the bottom, and the solute will travel depending on its solubility within that. Affinity chromatography is slightly different. We use that to separate proteins, okay, so much larger molecules. And we use a solid matrix or a gel column, okay, and it's created with specific molecules that are bound to the matrix or gel. Okay, so our matrix or gel would be down the side here. These are the specific molecules, okay, these little things here. And what would happen is that we would put our mixture in. If we want to separate some proteins, we'd put our mixture of proteins in, and we'd put them through this solid matrix or gel column. And what would happen is the target proteins within the mixture would have a high affinity for these little specific molecules here. And these pink ones in this instance would be the target proteins. They're the ones with the high affinity. And you can see them binding and becoming attached to those specific molecules as the mixture passes through the column. And then at the end, they would then wash out the whole column there. And what would happen is the non-target molecules with a weaker affinity for these specific molecules here would then be washed out, okay? And we're left with the specific proteins that we were targeting initially. Gel electrophoresis. So this is a process which is used to separate both proteins and nucleic acids, okay? They can use it for DNA or for proteins. And what they do is they put a charge through a matrix gel, and that electric field that is created will mean that those molecules will move through the electric field, and they will be separated by their size, their shape, their charge, depending on which type of gel electrophoresis you do. Now, there are two types. There's the native gel electrophoresis, which separates the proteins by their shape, size, and charge. Okay, so by the three elements there, they do not denature the molecule. Okay, they do not denature the molecule. That's a key thing to remember. So the native gels will separate them by shape, size, and charge. And because the proteins are not denatured, they will maintain their shape, their size, and charge, so they can be separated by those means. There's a secondary method, though, called SDS-Page. Now, SDS-Page separates the proteins by size alone. Now, how does it do that? Well, what it does is it will give all of the molecules an equally negative charge, and it will denature them, which separates the proteins, okay, by size alone, because we will have denatured them, will have caused all of those bonds to break, and the protein will have unraveled. And because of that, it'll have lost all of its ionic bonding and so on, and then we'll have an equally negative charge throughout, which means that it will not be separated by charge either. Okay, so we've got native gels, separate by shape, size, and charge. They do not denature the molecule. And we've got SDS-Page, the alternative method, which separates the proteins by size alone. And the reason it can do that is because it has an equally negative charge and it denatures the proteins. And then we can separate them on the basis of size. And you can see in our gel electrophoresis equipment here that it will travel from where it's input here at the top, it will travel through, and we'll see the larger molecules won't travel as far as the smaller molecules, so we can separate them by size on that basis. Okay, so that electric field that goes through there causes them to migrate through the gel there. Isoelectric points, or IEPs as it's shortened to. So proteins can be separated from a mixture using their isoelectric points. Okay, the isoelectric point is the pH at which a soluble protein has, and this is a key thing to remember, no net charge. And at that point, it will precipitate out of solution. It will appear out of the solution. If the solution is buffered to a specific pH, only the proteins that have an IEP of that specific pH will precipitate, because the pH, we will have that pH gradient, right? Because the solution is buffered only to that one pH. We won't have the gradient that you can see in this diagram up here. So proteins can also be separated using their IEPs in electrophoresis, the process we just had a look at before. Soluble proteins can be separated using an electric field and a pH gradient. Okay, we talked about the pH gradient there, and we've got our electric field there. Now, the way this works is that the protein will move through the field, it will migrate through the electric field and the pH gradient until it reaches its IEP and the pH gradient, because at that point it has no net charge. And we can see that in our little diagram here, okay? we have got a positive charge in these ones, negative charge in these ones, and they migrate through the field, negative going towards positive end of the field and positive going towards the negative, and they'll continue through until they get to the point where we have no net charge, okay, where it's equal positive and negative. And at that point, the proteins are going to precipitate, they're going to appear out of the solution. Okay, and that will be known as the isoelectric point of that protein, the point at which it precipitates. Okay, so you just look out for where in the pH gradient it precipitates, and that's its IEP. Detecting proteins using antibodies. So we're going to look at immunoassay techniques. Now, these are used to detect and identify specific proteins, okay? We might have a blood sample or something similar, and we're looking to identify a specific protein. It might be the antigens for a particular virus or whatever. These techniques use stocks of antibodies with the same specificity, which are known as monoclonal antibodies. Okay, they are clones, they are copies of the same antibody, and it's just one antibody, it's one specific antibody, which is why it's monoclonal. An antibody specific to the protein antigen is then linked to something called a chemical label. Okay, this is a chemical label up here in this diagram. The label is often a reporter enzyme, okay, an enzyme, which will produce a color change within the sample. Now, the reason why it will produce a color change within the sample is so that it can indicate to the people carrying out these little assays of tests which ones have a positive result, which ones have the protein of interest. But as well as producing a color change, sometimes we can use chemiluminescence, fluorescence, or other types of reporters within these assays. And it says there at the bottom, in some cases, the assays use a specific antigen to detect the presence of antibodies. So basically like a reverse process. So instead of having the antibodies at the bottom here, we would have the antigens, and then the antibody would bind to the antigen. But we can see here in this process here, we've got the antibodies. These are the ones that are specific to the antigen or the protein we're looking for. When the protein comes in here or the antigen comes in here, it will bind to that antibody, and we get that antibody antigen complex that we learned about in higher human biology. Then we'll get our antibody, which contains a reporter enzyme, will bind to that, and that reporter enzyme will then break down a substrate. Okay, that's this white substance here. We add a substrate to it, and that substrate gets broken down by that enzyme to produce a colored product. Okay, that's what will cause the color change, and that colored product will indicate the presence of that protein, that antigen there within the sample. If that protein or antigen is not present, then it will not bind to the first antibody here, and this second antibody with the reporter enzyme will not be able to bind to it, and that will not cause the shape change in the enzyme which activates it to break down the substrate. Okay, so there will be no color change in that instance. Western blotting. So we talked about SDS page as a method to carry out electrophoresis earlier on. We were separating by size alone. Now, once those proteins are separated from the gel, we can then transfer them or blot them, that's why it's called Western blotting, onto a solid medium, okay? So we're going to blot, this is the SDS page gel here, we're going to blot that onto a solid medium, okay? Then what we're going to do is we're going to add a specific antibody, okay? You can see those little Y-shaped antibodies there. They have the reporter enzyme attached to them. You can see the little orange E there, that's the reporter enzyme. And what that will mean is when we are looking for a specific protein from the SDS page, that reporter enzyme will break down a substrate to cause a color change, and that will then give us this little color here. In this case, it's a blue-violet color, okay? But you can see some of the other samples here are colorless, which means they did not have the protein of interest, okay? So we use a mixture of techniques there to find and to separate our proteins. Okay, moving on to microscopy. So 2 types of microscopy you need to know about for your exam. You'll need to know about bright field microscopy. That's probably the one that you've done before in class, where you've used a light microscope. You can see from the diagram, you've got a light source beneath the stage, and that shines up through the slide that you're using, and you'll be able to observe what's on the slide. Now, usually this is used to observe whole organisms, so maybe some bacterium or some sort of smaller organism, parts of organisms, or thin sections of dissected tissue or individual cells. So that those cells of small intestine and kidney and muscle and and blood and so on that you might have looked at in class, they will be all part of bright field microscopy, okay? So whole organisms, parts of organisms, or thin sections of dissected tissue or individual cells. Fluorescence microscopy uses specific fluorescent labels to bind to and visualize certain molecules or structures within cells or tissues. So again, we can see in the picture here, the nucleuses are very clearly coloured there. They'll stand out with that fluorescent sort of blue. We can see some of the cytoskeleton within the cells as well, and we can see the edges, the membranes as well of the cell. Okay, so there's lots of features there that stand out that are very striking and visual because we've used these fluorescent labels. And it means that if we're looking for a particular structure or a molecule within a cell, we can see it much more easily than we could do normally. So that's the purpose of fluorescence microscopy. And so moving on to aseptic technique and cell culture to finish with. Aseptic technique eliminates unwanted microbial contaminants when culturing microorganisms or cells. It usually involves the sterilization of equipment and culture media by heat or chemical means, and the subsequent exclusion of microbial contaminants. Okay, so again, you can see a couple of the techniques in the picture here. So we're using heat here. Now, the heat is good because we can use that on the inoculation loop, which we use to transfer our pathogenic organism that we're moving about. And we can also use that heat to help sterilize the air. We'd probably use some sort of antiseptic spray all over the desk and the other equipment as well. We would heat the neck of the tube as well before we moved our bacteria or whatever it is that you're culturing, and we would use these techniques to help keep a sterile environment. Okay, one that is clear of any contaminants, because we do not want anything getting into our samples, or obviously anything from our samples getting out either. A microbial culture usually started by an inoculum, okay, of microbial cells, and now we put them onto an agar medium or into a broth, which has the suitable nutrients that are appropriate for whatever it is that you're growing, okay, whatever it is you're culturing at the time. There are lots of different culture media that exist, and they promote the growth of specific types of cells and microbes, okay? So you would have a culture medium which is specific to whatever it happens to be that you're growing, okay? Animal cells are grown in mediums that tend to contain growth factors from serum, and the growth factors are proteins that promote both cell growth and proliferation, okay? Because we want those cells to grow quickly, and we want them to proliferate and to create many of those cells quite quickly. So those growth factors are essential for that. Okay, lastly, cell culture and counting cells. So when we have cells in culture, we can use primary cell lines, which tend to come from the tissues of the organisms or directly from the organisms that we're studying, but they can only divide a limited number of times, okay? Whereas if we take tumor cell lines, they can perform unlimited divisions, okay? They don't necessarily have that sort of cell specificity that we might be looking for, say if we're testing a drug or something on those cells, but they will divide indefinitely. They have an unlimited division. So that is useful for us when we're carrying out certain studies in a lab, whereas the primary cell lines are more useful for something that's a bit more specific to a particular tissue or cell type. When we play out a liquid microbial culture onto solid medium, okay, sometimes it can be difficult to count how many bacterium, individual microorganisms there are. So what we do instead is we count colonies, okay? We call them colony forming units. So we count those, and you can see from the image on the right there, some colonies have formed on that particular plate. And then from that, we can then estimate the density of the cells in the culture, okay, based on the number of colonies and the size of those colonies that we count. Sometimes, however, that plate will be absolutely overwhelmed with microbes, and the only way for us to get a suitable cell count is to go through a series of serial dilutions. Okay, and that's where we dilute our culture by a factor of 10 each time, and we continue to do that until we get to a point where the colony count is sensible and reasonable. We get to the point where we can actually see distinctions between the colonies. When we have cells within a liquid culture, we can use a hemocytometer to estimate the cell numbers. Now, we can see the hemocytometer grid here, just in this microscope. Okay, and we can see what it looks like when it's magnified there. Okay, that's useful to know what that looks like because that does come up in the exam from time to time, and we'll talk through how you work one of those out in a moment or two. Now, when we're estimating the cell numbers in a liquid culture, we have to be sure that we are only counting the live cells. Okay, we call that a live cell count, and the way we do that is through something called vital staining. So, vital staining is required to identify and count viable cells. Okay, that's the ones that are actively growing or dividing within a sample. And what we do is we add a stain or a dye to the cell culture, and then that will be taken up by the dead cells. Okay, it will move into the dead cells, but not by the living cells. And therefore, the ones that have taken up the stain or dye, we don't count, and we do count the ones that haven't. That would be a live cell count. Okay, if we only count the unstained ones. And we can see again, bottom right, an image there where we've got a dead cell amongst all the live cells within a culture there. Now, how do you actually calculate how many cells would be in a, a culture based upon our hemocytometer grids? Well, when we look at it under a microscope, now it obviously doesn't look exactly like this. This is just a diagram, but this is how it might look in your exam. So a question like this might come up, okay? And it says the grid is 0.1 millimeters in depth and calculate the number of neurons in 1 centimeter cubed of the culture. Okay, so there are certain steps that we should take when we see a question like this to work out how many neurons there are in that particular culture. So first of all, we should calculate the volume of the grid there. Now, hopefully you guys remember from your maths how to calculate volume, so length times breadth times height. Now, height on this occasion will be replaced by depth, the depth of the grid. So the length, and what we also have to do is we have to look at the units and maybe do a conversion here. So it wants the answer in the number of units in 1 centimeter cubed. So what I would do first is I would change those millimeter measurements into centimeters straight away. And that would mean that we are doing length times breadth would be 0.1 times 0.1. Okay, that gives us our area, which is 0.01. Then we times that by the depth. Now, again, that's going to be 0.01 because we're converting it into centimeters. So that's going to be, when we multiply that as well, that's going to be 0.0001. We're then going to count the number of cells. Then we're going to divide the total volume by the grid volume. So it'll be 1 divided by 0.0001. And that's going to give us 10,000. So essentially, this grid can fit into 1 centimeter cubed 10,000 times. So in terms of being able to estimate the cell number, that has given us a good clue. 10,000 of these grids fit in the total volume that we need here. So next we count the number of cells, okay? In this case, it's asking for neurons. So we count the neurons. There's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 within the grid, okay? You'll notice there's a few out with the area that we're concerned with. We can ignore those, okay? We're just going to be concerned with the 11 that are inside of it, okay? Now, if there are 11 in this grid, and we know that this grid goes into the total volume 10,000 times, we're going to do 11 times 10,000, and that will give us an answer of 110,000 neurons in 1 centimeter cubed of the culture. Okay, there's loads more of these in past papers and so on if you want to go and practice them a bit more, but that's how you work that out. Okay, folks, so that's us reached the end of topic 1.1 of the Advanced Higher course. I hope that was useful for you. Let me know in the comments if there's anything I missed or anything you want to know more about. But we'll see you in the next lesson for 1.2. Thanks, folks. Class dismissed.